Take it away, Neil.
Great. Well, thank you for that, Holly. Just one very quick addition for the Starfish Dash. If you're not in San Antonio, you can participate virtually pretty much anywhere in the world. So you don't have to be in San Antonio to be part of the fun. But I just wanted to, my name is Neil Parker. I'm the executive director here at Chromosome 18, and I have had the pleasure to serve with the organization actually coming up for 6 years next month. So is that been quite a while, but that pairs into, in comparison as to how long Dr. Janine Cody has been with the organization. She started it kind of 32 years ago when her daughter Elizabeth was born. So the the Chromosome 18 Clinical Research Center is actually celebrating its 31st year this year. And we're going to have the opportunity to go through with Dr. Cody to kind of see all the magic that works over there. So without further ado, I'm going to give it over to you, Dr. Cody.
Well, once it was really loud. So can you hear me? I think, did we test my sound? I can't remember. Anyway, welcome. Glad you're here. If we were here in person, you would be driving up to the Health Science Center and coming into the building that is the medical school. You come in the front door and come up the elevator to the 5th floor, and it's way down at the end of that hall. You come up the elevator and come all the way down this hall. These are this is the Department of Pediatrics within the School of Medicine. And the offices and labs down this hall and the next are all members of the department where their research happens. And so here at the end of the hall, we have the Chromosome 18 Clinical Research Center, which is easy to spot because Annis has created this amazing board. Actually, there's 5 of them showing all these amazing smiling faces. Families have sent the pictures for the purpose of putting on the bulletin board for everyone to see. So I love coming in every day and seeing this. So if we can move over here, I can tell you a little more about what we do. Now that you know why we do it, so on the smiling faces. Just a little background though. Chromosome, you probably know about Down syndrome. Down syndrome is actually caused by 3 copies of chromosome 21 or trisomy 21. But you can actually have missing or extra pieces of any of the other chromosomes. Most are compatible with life, but ones that are involve chromosome 18. And so you can also have 3 copies of chromosome 18. That's the second most common chromosome abnormality after Down syndrome. However, babies with trisomy 18 are usually quite medically fragile, and about 90% don't live past their first birthday. Most of them die very shortly after birth. And because it's more common and more well-known, and been recognized for a longer time, other groups have been really interested in trisomy 18, so we haven't focused on that quite as much. However, no one has paid any attention to those unique and amazing long-term survivors over the age of 1. And so that's what our focus has been. And so we've enrolled people with trisomy 18 who are at least mostly over 4 years of age. But there's, as I said, all kinds of other chromosome 18 abnormalities. And when you look at the picture of the chromosome, if you look at any of these diagrams of what the chromosomes would look like under a microscope, you can see somewhere along the length there is a constriction. Chromosome 18 is always right, not exactly in the middle, so it looks like there's a short arm and a long arm, and the convention is to always show it with the short arm at the top and the long arm at the bottom. The short arm is called the P arm, and the long arm is called the Q arm. So it's just what it is. So since I've already told you there's all variety, we've kind of grouped them into 4 other groups. And so someone right here, you can see has 18q minus. And so this person, people with this have a deletion of any size from any region within the long arm of chromosome 18. So you can imagine there could be a lot of variety, and in fact there is. We have almost 500 people enrolled, and no 2 people have exactly the same deletion. By the same token, people can have a missing piece of any size from any region on the short arm, the P arm, and it's called 18p monkeys. And again, there's a lot of variability because of that. People can also have one of their chromosome 18s form a ring. And by definition, that means that the caps called telomeres of the ends of the two ends of the chromosome are missing. And so the chromosome ends are sticky and form a ring. And so usually there are also genes missing from both ends. But again, the number of genes, the size of the deletion from each end varies. So each child with, or each person with ring 18 is very different from another. And what I should have said was 18q minus and 18p minus occur about 1 in 55,000 births. Ring 18 is about 1 in 300,000, I think 325,000 births. And the most rare is tetrasomy 18p. It's 1 in about 600,000 births. And it's unique in that individuals with tetrasomy 18p have an extra chromosome. However, that chromosome is made up of 2 P arms. So there's 1, 2, 3, 4. So it's tetrasomy 18p. And so this group is pretty uniform, actually, because they pretty much all have exactly the same duplication. And the families who are enrolled here come from all over the world. This is now a couple of years old, but serves the purpose to illustrate where the families who are enrolled in the research come from. And families enrolled in the research don't necessarily have to be members of the Chromosome 18 registry. This is an entirely separate operation from the Chromosome 18 registry, where the registry, the support group, is interested in family services, in community building, in fundraising, and advocacy, education, having conferences, that sort of thing. Where on the research, we do research. And in order to do research, you have to sign consent forms to document that you understand what the research involves. And so all our consent forms are in English. And so that's one of the obstacles why there aren't people from lots of other, even though they're from all around the world, there are not a lot from huge parts of the world. And while it'd be really easy to translate our consent forms in other languages, the problem is we do lots of mail out sort of surveys or email or regular mail surveys that are standardized surveys, like for autism and behavioral sort of things. They're only normed for English speakers. So we could gather the data from those people, but we can't use it. We couldn't publish it. So, so that's one obstacle. The other is we need a blood sample, which I'm going to show you about later. And that needs to get to us within 3 days, and which I'll explain when we get to the lab. And so that's the real obstacle for getting blood from wider regions of the globe. And many families have managed to do it. As you can see, we've got families enrolled from New Zealand and Australia, Indonesia, South America, all over Europe, and some in Africa. So they really, we really do try to include people as best we can from anywhere in the world. So, and the team here is shown here. It's myself, Dr. Hales, medical director, is now in Hershey, Pennsylvania, and John Gelfond is associate director. And so he's a bioinformatician who's here, MD, PhD bioinformatician. And the research staff includes Annis Hill, who is working from home today. So you won't meet her, Patty Bird, who doesn't like to do tours, so you won't meet her today. And Alex Brewer, who's not in today, he's working from home. So, but I don't let them talk anyway, so it's all right. So that's our staff, and we have 2 administrative staff, Gloria Matthews and Linda and Sarah. And then we have a whole group of investigators. They're all faculty here at UT Health from all kinds of specialties. And so you can imagine that because there's 263 genes on chromosome 18, someone with a duplication or deletion of a chunk of that chromosome, depending on which chunk we're talking about, they could have congenital heart disease, they could have growth issues, they could have autism or not, or they could have hearing problem or vision problems. Not everybody, it's a long, long list, but not everybody has everything because they have duplications and deletions of different genes. But because in total, there's this huge variety of potential issues, we have this huge team here that includes pretty much all the medical subspecialists that you can think of. So, you know, neurology and immunology, sleep medicine, psychiatry, physical therapy, ophthalmology, otolaryngology. And as we've learned more, we've included people with very specific expertise, like Dr. Meyer, who's Dr. Meyer, whose office is right here, who's expert in platelet function, and Dr. Maria, who's expert in bone development. And as we have started to identify some of the genes or key regions, that allows us then to start thinking about what's the basic science that we need to learn to understand how to upregulate or downregulate those genes in order to implement a therapy. And in order to do that, you need cell-based systems, you need mouse models. And so now we have collaborators who are expert at that. So Donna Lehman does the cell-based assays and cell development. Let's see, where's Junie? Does electrophysiology, laboratory, creates mouse models, as does Georgiana Gould, who's also here. So it's this huge, huge team of people, all who are volunteers. I spend most of my time bugging people. Please, review that record that you promised or review that draft of a paper, that sort of thing. At this point in time, we don't have funding to help support the clinical collaborators. We have in the past, but we don't at the moment. But they're committed to this project. So they do it because they realize this is a really important and interesting project. Let's see, show you around just a little more and interrupt if there's any questions. Here's our office suite. So I could show you here, pictures on the wall. Anna sits here, my office is back there, Alex is back here. So similar suite across the hall with our administrative staff. And the lab is down the hall and around the corner. So we'll walk down there. And I think usually Neil says a few words at this point while we're traveling.
So as Dr. Cody moves down the hall and to the lab, a couple of things that I just wanted to share with everybody. Number one is that we actually have 4,000 members across the USA, and we have about 2,000 members around the world. So, but everybody is able to join in any of the programs that we're part of, and so whether it's the new syndrome groups or whatever, is that everybody is welcome. So, as Dr. Cody has arrived at the lab, I'm going to pass it back to her.
Okay. Are you ready, able cameraman? Probably is our cameraman, just so you know. All right, so when families enroll, as I said, they have to sign consent forms. And of course, they also have to send us documentation that their child actually does have a chromosome 18 abnormality. We have had people try to enroll whose genetic report said had the number 18 somewhere in it, but it really wasn't a chromosome 18 abnormality. So, and sometimes that's what the family picks up on, or their doctor even picks up on. And so we need proof, first of all. So we need that proof. We need the consent forms, and we need a blood sample, as I already mentioned. So families who already have sent those two things in, then they will get a blood kit with all the tubes necessary. And this will take a lot, but we get blood from the affected person and both their biological parents. So they really only have to send a third each. But we do 3 different things with this. So that's why there's so many tubes here. And let me show you a little bit about what we do. The first thing we do is, or one of the things we do, is isolate DNA from some of those tubes of blood. And this is piece of DNA, and and you see the white string swirling in there, that's DNA. And that's the second to the last step, because it doesn't work very well to try to analyze it when it's in this form. So this next would go in the centrifuge over here, get spun down, and then taken up with a hydration solution. So we would go into solution so we could do analysis. But you can't see it then because it's in solution. So this is why we show you this. So we have DNA on all the participants who have a chromosome 18 abnormality, as well as both their biological parents. And what we used to do, what used to be done was chromosomes were visualized under a microscope, and you could see images kind of like what I showed you on the board. And so you can imagine if you took scissors and cut off a chunk, okay, you say, yeah, it's different, it's missing a piece, but you can't tell from that what genes are missing or exactly where a breakpoint might be on a molecular level. So we need to do molecular analysis. So let me show you some of that. And this is, so what I'm going to show you is actually just a partial screenshot from the software program that we use. So what we do is the DNA along with special solutions and oh, good stuff. There we go. DNA gets put on a what looks like your average little 2-cent microscope slide, but it's not. This is a $600 microscope slide. And what is on here are grids of about 250,000. Actually, there's 4 grids on here, each with about 250,000 manufactured spots of DNA that are unique to a very specific place within the human genome. And you put the DNA on there, and you go through several processes. Takes a couple days. Goes in a laser reader, and you get data that looks like this. This way. And again, this is just a partial screenshot from the software. Otherwise, it's just like overwhelming screen to look at. So I'm just trying to highlight what's important. And so what you have here is, here we have our picture of the chromosome here. Every single one of these dots, you can see some are black, some are red, some are green. Every one of these dots is a data point that is specific to one of those manufactured dots on the slide. And what you can see is all the dots around this zero line. Here's a zero right there. That's where this person has 2 copies of the chromosome. And you can also see this is a really noisy kind of experiment. There's red ones here and there, and there's green ones. But what counts is when you have contiguous ones in a row that are either showing a duplication or deletion. And that's what the software measures, it calculates. And so what it's showing us is that right here in this region where it's showing up as a red bar here, where all those little dots are green, or a majority of them are green, that's where this person has a deletion on chromosome 18q. The long arm for this person has 18q minus. And for me personally, what's totally amazing about this is my entire length of graduate school, this is what I did, 1 dot at a time, each one taking 3 days. And for, at that time, a couple hundred people. And this is 40,000 right there in 3 days. I mean, we got this technology, and I just felt like I died and went to heaven. This was so much higher resolution than we ever had, by factors of thousands, and not to mention the days saved. So while that slide might be super, super expensive, it's a whole lot less expensive than a decade worth of somebody's salary who still can't get that resolution of data. So what you can do in the software is, we want to look at this region. We want to know what genes is this person missing. So what we can do is zoom in. And so if you zoom in to this blue box here, this is what you would see. Again, you can see it's still pretty messy, but you can see a distinct region right here, where all the dots are shifted left, and that's where the person's deletion is. But what's also showing up here is where the genes are. So what's in gray, these little bars in gray are the various genes. And some of them are kind of spread across the screen. So they're kind of, otherwise they'd be on top of each other almost at this resolution. So they're spread across just for readability. But in reality, they're really one after the other going this way. And you can zoom in and zoom out, and you can determine that this person's deletion does not include this gene, but it does include that gene. And at the bottom, you can see it does include this gene, it does not include that gene. So we can know exactly which genes this person has one copy of instead of two in 3 days. Oh my gosh, it's amazing. This technique is now standard in clinical diagnostic labs. When we started doing this, this was a research technique, not a clinical technique. Actually figured it out the other day, and now I've already forgotten. Where we have 40,000 data points here, a standard clinical, high resolution clinical array that most people get when they're diagnosed today has only 10,000. So it's really, really good. It's not this good. In most cases, it's good enough to pass. It gives us enough information. So, but there's a problem with this. Not everything's technique is perfect. So we need to do more than one thing. The question becomes in this, I should get rid of that for a minute. Where is this piece right down here that this person has 2 copies of? Is it stuck right there on the chromosome? Or is this off somewhere else stuck to another chromosome? Or floating freely, which really wouldn't happen because it'd be lost. So we really only have 2 choices. It's either stuck up here or it's on a different chromosome. So that's what we do. Another part of the blood sample is we prepare a nuclei prep for looking at chromosomes under the microscope. However, we use special techniques, not just light microscope looking at black and white bands. This is called whole chromosome paint, and this is the same sample. The first one is DNA. These are the chromosomes from cells. Visualized under the microscope and treated with a special fluorescent dye that's specific for chromosome 18. So here's all the other chromosomes. That's probably chromosome 1. Where's the itty bittiest? That's probably 21. Oh, here we go. Probably 21 right there and there. Here's chromosome 18 with the fluorescent dye. What's important here is you can see a couple of things. First, each of these chromosomes is entirely covered with dye. So they're all chromosome 18. There's not some other chromosome material stuck in there somewhere. But you can also see there's no green dye anywhere else. And what we really do is we look at about 50 of these to make sure they're all the same. But that does help us answer a little bit of the question. It gives a hint that that piece at the bottom was really stuck up higher on chromosome 18. So it's kind of a looped out sort of deletion. But we can do one more thing. We can use really fine detailed probes. And in this case, from the same person, we have 2 different colors, green and red. And the green probe, you can see here, is for the centromere, that waistband on the chromosome. It marks the chromosome 18 centromere or waistband. And you can see one is marked here and one is marked there. So that's where the 2 chromosome 18s are. The red fluorescent dye attaches to the very bottom end, the bottom of 18q, and where that person had 2 copies. And here they are. This one is attached to the same chromosome 18 as is that. So that proves that this person does have an interstitial deletion, and that full piece of chromosome at the bottom is not stuck somewhere else. So that's what happens with the second part of the blood. The third part of the blood goes into a tube like this, which I'm just going to look up the price. They used to be 15 years ago. They used to be, when I was doing the ordering, they were about $4 each, so they're probably $8 now. Put the blood sample here, put it in the centrifuge here, spins it around. All the red cells go to the bottom. The white blood cells go to the top. It's the white blood cells that still have a nucleus and are able to divide. And so there's a little band of white cells, and those white cells are pipetted off of here. and put in a flask like this, tissue culture flask, along with media, which is growth media for cells. You can see it's nice brick red. And so we put them in here. And if that's all we did, and put them in the incubator at body temperature, which I'll show you in a second, they would double a couple times and they die. That's normal. Cells don't live forever, unless of course they're cancer cells, which is the fundamental problem with cancer. So what we do is we put Epstein-Barr virus in here, and it transforms many of the cells, and so they will keep dividing. So they will, so they'll start growing. So we put the virus in here with the white cells and the media, and then they go into the incubator right here. This is at 37 degrees, so that's body temperature. And I can show you some of these. So because these particular type of cells, white blood cells, they, does it help to have the paper behind it? They grow in clumps. So you can see them floating there. Those aren't individual cells. Those are clumps of cells floating. And so, and the other thing you can see here is this isn't bright red, is it? That's because these are happy cells metabolizing all the nutrients in the media and making it change color. And so that's how we know when we need to add more media, because there's enough cells. And there's an art to this. We couldn't just pick up a little bit of white cells and put them in this giant flask with a whole bunch of media. They would die. They like to have this certain, not too much company, not too little company. There's an art to this. And Patty is expert at this. And so what she will do, probably maybe later today, maybe tomorrow, is she'll add a little bit more media to this. And those cells will grow. And, 3 or 4 more days later, she'll add more, move it to a bigger flask. When this gets about to here, then she freezes them down. Well, if you were here in person, I'd let you look at these under the microscope, and this is what you would see. So I realize not nearly as exciting, but so like here's one of these big clumps of cells. And so these are all individual cells. And so what Patty will do when she feeds them is she'll put a pipette in and or mix them up, stir them up a little bit. So this breaks up because the cells in the middle can't get to the media. So these are the ones that are really most healthy. And you can see there's different size clumps when you start breaking them up. If these look like little grapes, we are golden. If they look like raisins, you are in trouble. And so you got to pay a little more attention to them. But that's what they look like. They're just little dots. And I wish you were here to look at them under the microscope. So as I said, when this gets to be, full, about to here, 100 milliliters, maybe 80 milliliters, then we freeze them down. And then we can thaw them and use them at any other time. So let me put these, so let me show you a little bit more then also about the lab. So all the work that we do when Patty adds more media to cells or even sets up the cultures, all that work has to be done in a biosafety cabinet, which we just call the hood. And this has it's a laminar flow hood. It's got a curtain of air right here that keeps air from the room from going in there. So it keeps it sterile in there. There's not bacteria floating in there. If one bacteria got into the culture, they double every 20 minutes. So do the math. By the next morning, you've got probably 1 million cells in there of your bacteria. Blood cells double every 24 hours. So if you've got 1 million cells, you just have 2 million the next day. You get one bacteria in there, the next day it's overrun with bacteria. So we've got to make sure it's sterile. So all the work done in here is sterile. All the pipettes that are used are all disposable. As much as I hate to use lots of plastic, that's the way you got to go to make sure it's all sterile. So all that work happens here. when, like I said, when the cells are grown up and she's got 80 or 100 mils in that big flask, she spins them down in the centrifuge and takes them up in special freezer media that prevents ice crystals from forming in the cells. And then they go into little suspension in that freezer media, go into these little cryo vials like this. And the cryo vials then get stored in liquid nitrogen in two forms. Little vials get into a cane like this, or into boxes like this. And let me show you, they go into liquid nitrogen, and this is our retiring tank, but it's a lot easier to open than the big ones. So it's why you get to see this. So this is full of liquid nitrogen. If you were here, you could see that it looks like water down in there. Clear. So there are 6 canisters, each with about 30 canes, and it's really, really cold. So I really don't want to hold on to this too long, but you can see that there's all these canes filled with samples. And we have three, and I guess the other 2 are in the other lab, I guess. We have 3 of these big tanks and plus the little one. And so we freeze down at least 3 vials by every single person. Every person with a chromosome 18 abnormality and each of their biological parents. And 1 vial goes in this tank, 1 vial goes in each of the other tanks. So these are just giant thermoses. So if they spring a leak or something happens, we haven't lost everything. We always have 2 backups. These obviously a lot bigger, and there are little canisters. In here, there are 6 of these things, which is empty, is heavy. That's why I use the little one. And each one of these has boxes in it with all these vials. So it's the same exact thing, only bigger and heavier and harder to get the column, the canister out and put back in. That's why we use the little one. I think that's it in the lab. If there are any questions while we're in here, or if not, I'll go back to my office and we can chat then. Are there questions in the lab that people have?
No questions at the moment, Janine. But if you want to make your way back to the lab, I'm sure there'll be a few questions by the time you get back to your office. So thank you very much indeed for doing that. And I really think it helps everybody get a visual on kind of like the amazing work that goes on there. So Amelie, thank you for being the camera person. And as Janine makes her way back, I'll just cover off a couple of things. So as Holly said earlier on, if you do have any questions, please just type them into the chat. You're also more than welcome at this stage to turn your video on if you want to, and you can ask Dr. Cody questions in person. But I'm going to pass it back to Holly, who will finish up for us with Dr. Cody.
And I just popped a link into the chat box for the research study if you're interested in knowing more. That is in there, as well as the Starfish Dash. And I'm about to post our resource page as well as the clinical management guides. So go ahead and copy those. I'll also send them in a follow-up email after the tour is over. But again, feel free to unmute yourself, turn your camera on now if you'd like. This is going to be a time to talk with Dr. Cody. She is super nice, super knowledgeable. So feel free to turn your cameras back if you'd like. I'm going to pop a few more things into the chat. I do see one question. So when Dr. Cody gets to her office, we'll go ahead and start with that one. Thank you, Maria, for your first question. Let's see, just pop in a few links in the chat box.
So Janine, just whilst Holly's doing that, is that I know there's some questions that we often get asked, and we've got some very specific questions that are kind of coming in. So Maria asked us, why is the parents' blood tested, and has there been any correlation in the parents' blood samples?
Well, let's see. The parents are really only used for reference to compare to the child. And back before microarrays, we had to have the parents. It just the method required comparing all those little dots to the parents. It's not really required anymore. However, one of the things that we've become interested in, let me see. I mean, so we've used the parents also to compare for gene expression to see if there's differences. We are also working on a project to try to understand some of the unique things that kids with chromosome 18 have. For example, let's say like congenital heart disease. Really not very many have congenital heart disease. So the question is, why this child and not that one who has the same genes deleted or nearly the same? And in many cases, we have a good idea which gene causes which feature of the condition. And so one theory is that the child had the few people with congenital heart disease, they have a deletion of the key gene associated with it, but they also have a mutation in the other gene. And it requires both things in order to have congenital heart disease. And so that would imply that one of their parents is a carrier, meaning has a mutation in that gene that puts them at risk for this. But of course, they don't have it because they're a carrier. And so the way to test that is to see if a change in the child is indeed inherited from the parent. So so it's only really for reference to help us really understand what's going on with the child. We don't do routine analysis, and we don't have medical records on the parents, so we don't, we can't really go anywhere with any genetic information we might have about them. So it's all in comparison to the child and what they have. And it's all for the purpose of understanding the chromosome 18 conditions better. I hope that, and that if that didn't answer your question, ask again.
She said, thank you for answering, Dr. Cody. And we always like to ask this question: how does the research help individual families and the chromosome 18 community as a whole?
Mostly in the most direct way, of course, it's the management guide. So all the papers we've written over all the years, and actually a lot of data that's not in papers is put in the management guides to help families understand, and their physicians understand what are the important things to monitor, what to watch for, what to evaluate. And in a way, kind of what to expect, even though that there's a huge amount of variability, it still helps to have some idea what the potential might be.
And those management guides are such a useful tool. As I've been getting to know all of the families, so many of them say these are key to take to our doctor's appointments, to share with our doctors. So that's awesome.
Yeah, I realized at one point, that nobody's pediatrician has time to go look up 6 papers one about, I don't know, seizures, and one about hearing, and one about, and even a review article. They don't have time to go read all that. They need something now. I need to know what I need to do today. And so that's what we tried to create, is take all the papers and we synthesized all that information so they could have something that just takes them, seconds to figure out.
And we just did a Facebook Live with Dina, a genetic counselor, and she pinpointed those and said those are so crucial to take with you to your appointments. And where can people find those guides?
They are really housed on the Research Center website, but there's links on the registry website as well. So,
and I just popped it into the chat too. Oh, let me pop it into the right chat. There we go. Awesome. Does anyone have any other questions? Great time to talk with Dr. Cody. Thank you again for sharing this time with us.
Of course, the goal, don't forget, the goal, which we're still a long ways from. I mean, the management guides are huge, but the goal is to be able to develop very specific treatments that would help improve, I don't know, reaction times, I don't know, bone health. I mean, all those things that might be, that are to treat them in ways that are very specific for the origin of the problem that the person has. I mean, we all know people, I have a good friend who says he could take a whole bottle of Tylenol and it wouldn't make a dent in a headache. It doesn't work for him. You know, most medicines out there, they don't work for everybody. They just work for most people. So what we're really talking about is really targeted things focused on the source of the problem for people with chromosome 18 abnormalities. And not just because there might be better targeted, but these are things there aren't treatments for in the first place. So, or treatments like depression or seizures, where you got to go trial and error on a whole bunch of things before you figure out which one works for you. And we think that by knowing exactly the genetic mechanism behind each person's type of depression or seizures or whatever, that will give us a better starting point of which medication to use, even if it's a standard one, or if we have to develop a new one.
And I have a question. I know we have our conference coming up in July. Can families join the research study there?
Oh, yes. Oh, yeah. Very good question. I'm glad you asked that. Yes. However, you might have figured out from listening to the tour, though, we will have blood drawing available, because that's a big obstacle for people. It used to be every pediatrician did it in their office, and that's not true anymore. They send them to Quest, and Quest won't do it because they're not doing the testing on it, on the sample, because we've tried that avenue. But you have to have contacted Annis, the program coordinator, and you have to have signed the consent forms, and you have to make sure that we have the chromosome report. And it's really good to have some other medical records as well. And so once all that is ready, and the only thing left, which is always the last step, is a blood sample, then you're prepared to give blood at the conference. But we need to know about it beforehand. Annis needs all that paperwork beforehand. So we have the right supplies there.
Definitely. That's a really good point. If you're interested, you know, I can put Annis's contact info in the follow-up email. They'll reach out to her, and then if they're coming to conference, be able to do the next steps there.
Yeah.
Awesome.
So we and just so you know, check-in for the conference is, let's see, it's a Wednesday this year. All day Wednesday, sometimes even the day before Tuesday, they're set up to do blood draws. And so lots of families do it before the conference starts. They come in a little early because they try to, and she schedules a time for families. It's not just you're just standing in the hallway waiting. So she'll schedule a specific time. So think about that as you make reservations, that if you come in just a little early, you can get the blood draw over with. But we'll draw blood through the entire conference if needed. We have some amazing moms who are nurses who all volunteer their time to help with that.
Well, that's great. Well, awesome. Does anyone have any other questions? Feel free to unmute yourself or type it into the chat box.
Dr. Cody, one question I suppose I would ask is that for people that don't know, is that, you know, what would you need to continue all of your work, and who kind of funds all of the research?
Well, you may have guessed that studying chromosome abnormalities just isn't the end thing to do. There are very, very, very few opportunities to even apply for grants applicable to this kind of work from the National Institutes of Health, which is the main government source of biomedical research funding. You know, probably every day I get a notice about some program announcement for our cancer research. You know, if you're doing I mean, I can't even tell you the last time I saw one that was applicable to chromosome abnormalities. Rare diseases sometimes, and so that's what we've been focusing on applying for those that are focused on rare diseases. But again, you know, they don't come around very often, and so that means that the funding has to come from other places. It's not we're not government funded. To keep enrolling families, to keep sending out surveys. This is a lifetime longitudinal study. Oops, I should have said that at the beginning. So we have no idea how are these people gonna age? Are they all susceptible to early XYZ, whatever it is? Something preventable if we just knew about it early enough and we're monitoring for it? I mean, we just have no idea. I mean, what we do know is for the vast majority, except for trisomy 18 and except for people who have other chromosome stuff going on, like also deletions or duplications other places, all the rest of them, we think, have pretty much a normal lifespan. And we don't know what health issues they're at risk for. And so monitoring people, sending out surveys, keeping in touch with them, collecting medical records on an ongoing basis, and enrolling new people, that takes staff time, at which and lab time, obviously, and that cost. So the bare bones cost is about $300,000 a year. But there's all these other things we haven't done yet. We haven't had ability to assess people with partial trisomy 18 or the long-term survivors with trisomy 18. We've only assessed, and I guess another thing I didn't really say is when there's funding, we bring in families for an assessment over many days by all those people whose picture you saw out in the hallway. So they see a neurologist and an ophthalmologist and an ENT and all those people, and neuropsychologist, of course. But that's only when there's funding to do that. And so we've only assessed, I think, 10 people with 18p deletions. And now we know, at this point, we know exactly what size deletion they have, but we need to assess more. And now we could assess adults. And so we could assess the adults and look back at their trajectory and see what's happened with them. We could learn so much more, but we just don't even begin to have the funding for that. There are other basic science studies we could do to look at the cells, and because one thing you can do now, you might have wished you hadn't asked me this question, but one of the really cool things that you can do now is we can take those blood samples, and even though it's a white blood cell, they've now, and you may know this if you follow any science in the press, you can now cause them to differentiate from blood cells back into more primordial stem cells. And then you can take those stem cells and differentiate them into neurons or oligodendrocytes or the blood cells from the brain. And so now we can ask, how are the nerve cells in the brain of someone with an 18p deletion or tetrasomy 18p, any of them, how are they different from normal? And so that costs a lot of money too. And that's some of those other investigators out there. We've got the ability to do that. We have collaborators who are expert in those things. We don't begin to have funding for that. So there's just so much we could do. So, and we're working, I mean, we're looking for opportunities for funding from outside sources every single day. I'm on all these lists that send in opportunities, and which I look at all the time, and for one that might fit what we need to do next. So anyway,
thank you. We can definitely tell you're
story
passionate
is,
about it.
have a team for the Starfish Dash, please.
Awesome. Well, does anyone else have any questions before we wrap it up? Are you excited for the Starfish Dash, Dr. Cody?
Oh yeah, I've been trying to rally support around here for being on a team, and people aren't answering. But you know, it's a few days out, and people make those kind of decisions last minute. So like everybody on that board got an email from me And they'll get another one, plus a few more people around here. So, so we'll try to get them to show up. If nothing else, they better show up because it's on campus. It's right
Yeah.
here. They got
And
no
like-
excuse for not knowing where.
Yeah, and like Neil mentioned, if you don't live in the San Antonio area, you can still participate wherever you live virtually. We're gonna
Join
ask
Team
people to post-
Liz. How
There
about
you
that?
go. And that link is in the chat box, but I will put that in the follow-up email as well. So we hope you guys can join us.
Yeah, that'd be great.
Yeah. All right. Well, I don't see any other questions. So Dr. Cody, just thank you again for sharing your time, your expertise. It's
Oh,
always a great tour. We always learn so much. And Neil, thanks so much for being on this call with us and just everyone for joining. We
Yeah,
hope you
thank
enjoyed
for
it.
joining. Thank you for being here. Thank you for your interest.
All right. Well, y'all have a great rest of your
Okay.
day.
All right. Thank you.
Thanks, everybody.
Bye, everyone.